One of the most common questions I get from my students is how to learn complex topics. Stuff like this, or this. And when I answer that question, I like to usually start off with an example. So here's a research paper. It's titled Temporal Lobe Epilepsy is Associated with Neuroinflammation, Extracellular Matrix Remodeling, and Synaptic Protein Alterations. That's a bit of a mouthful. And upon reading that research paper, a lot of people, myself included, will probably say that this is a complex topic. But remember this, complexity is largely subjective. If you hand this paper to a lawyer, for example, they would struggle through it. I mean, they don't have any background, but if you were to hand the same paper to a clinical psychologist, someone working in a neuroscience lab, or anyone in psychiatry, they'd have an easier time through it. It's the same paper, but two completely different experiences. So the question is, why does a psychologist have an easier time with this paper compared to the lawyer or compared to a layperson? Most people make the assumption that the psychologists are smarter, they know more about the topic, they're already familiar with the domain. That's correct. And it's actually the point. Let me explain. And it's worth digging into because us non-experts, we can do something about that, especially if we are trying to become an expert in this field. We don't have to stop at they're smarter and then just walk away and leave it at that. Let's explore that a bit more. So, we're correct. The expert knows a lot, and they spent years building up knowledge in their domain. But more than that, if you actually take a look at what's happening under the hood in the brain, they've got patterns and ways of thinking about that knowledge that make new information, this new complex topic, easy to learn and easy to latch onto. Their brain isn't doing anything special here. It's just doing what it has been evolved to do. It's naturally actually engaging in that process when the conditions are right. The expert schema, their knowledge, the way that they already think about that information and that prior knowledge create those conditions automatically. So this complex paper is actually super easy for them. And hey, maybe it's happened to you where people find something really, really hard, but you're finding it really easy. The part that most of my students miss is that you do not need years of expertise to access a similar process. You can run the same underlying approach their brain is running to, the expert's brain is running with your current level of knowledge. And you'll still be able to get to a level of meaningful processing on that topic that previously fell out of reach. Now, it's important to mention that you won't match the expert's depth because if you're learning complex topics, you are likely trying to become an expert yourself, right? But what you can do is you can close enough of the gap to the point where the topic doesn't feel like a wall. It doesn't feel impossible. As I mentioned, you won't outperform the expert, but the point is you'll do better than you probably currently are because most people, when it comes to learning these complex topics, I just find that they are leaving their learning capacity and potential on the table by not like setting up their brain up to the way that their brain wants to actually learn. So a framework that I like to teach that allows us to tap into how an expert is already thinking about a certain topic is what I call the 4-phase technique. And it's a variation of a technique that I share with my students. There are 4 phases, and each phase has their distinct technique. I'm going to explain each of the phases, and then I'll do a demonstration of all 4 phases on that research paper that we talked about, the temporal lobe research paper that we introduced in the earlier part of the video. Phase 1 is existing knowledge. Before you touch new material or that new topic, lecture, book, whatever it is about that complex topic that you're trying to master, you want to write down everything that you already know that's adjacent or related to that topic. This can be concepts, terms, experiences, intuitions, hypotheses, whatever you have here. Most people skip this because they assume they have no prior knowledge. In my experience, students always have more than they think. If I just sit in front of them and I'm like, you have to guess, they always produce something, right? So it's possible. And we're going to use a technique called blind mapping here. Phase 2 is existing patterns of thinking. So this is a step beyond just immediate knowledge. What you want to do is you want to start naming patterns or ways of thinking that you think could apply. So if you already think in a certain way at work, that's transferable. If you are a programmer and you debug code, you might know how to isolate variables and go through that process of iteration. If you cook, you might have a certain way of thinking about cooking. You know how to sequence steps. If you've done research in other fields, you know about research methods, and so you can think more like a researcher. You want to point to the patterns that you already use and try pointing that towards the new content. And one of the ways that you can do this is using analogies. Phase 3 is foreign knowledge. Now that you're starting to engage with the topic, you're going to inevitably encounter content and keywords that you don't know. And this is going to be for the majority of that topic. And so what we're going to do for this topic, this complex topic, is we're going to start having a search strategy. We're going to be isolating the keywords and the concepts that we're not familiar with. And we're going to lean on diagrams, videos, infographics, anything visual to help us explain this. Again, in phase 3, we're skimming and we're trying to connect slightly new information to what we already know. And we're slowly then moving into more unfamiliar territory. And we're going to use scaffolding here. Phase 4 is foreign patterns of thinking. And arguably, this is the hardest phase. This is a phase that a lot of people don't necessarily reach, but it's the most powerful phase. Some topics require genuinely new ways of thinking. So, for example, if you're familiar with research in economics or social psychology, the way that they do research in those fields might be very, very different from how they do research in clinical psychology, in psychiatry, in different domains. And so now you might need to be developing more of an awareness and appreciation of how a domain expert changes their way of thinking based on the domain that they're in. And this is where we're going to have to be thinking a little deeper and using interleaving here. So let's do a demonstration of all 4 phases with the research paper that I mentioned earlier in the video. So these are the 4 phases. Phase 1, existing knowledge. Phase 2, existing patterns. Phase 3, foreign knowledge. And phase 4, foreign patterns. And, before we even start diving into the actual topic at hand, it's useful to define what is a complex topic. And when we want to master complex things, what does that usually mean? It's just useful to have this defined. And when I have conversations with my students, it's specifically in their mind, complex things is when there's lots of like unfamiliar new terminology. They want to be able to use a lot of that new terminology. They want to be able to understand that weird esoteric terminology, like, in this situation here, I'm just scrolling a little bit. Gamma amino butyric acid, GABA receptors, like, they want to be able to understand like these keywords and be able to explain them. And in this paper here, potentially want to be able to understand like what is this even trying to represent, like this diagram or this figure. So that's what it usually means in that sense when they're trying to learn complex things. And these 4 phases, they try to capture holistically how we can approach learning complex content. But these 4 phases occur across weeks, months, years, right? It's not like you just do all 4 phases in one study session, therefore you're an expert. The reason I want to just take the time to mention this is to make sure the expectations are set well. To develop expertise, you need to spend time with experts and you need to slowly learn how to think like them. And obviously, the more time you spend with them, the more questions you ask, the more, if you're trying to become a researcher, the more research that you actually read and do, right? If you're at university or engage with, the smart people here, you actually start to develop that expertise. So, the video that we're making today is exploring a few tools that you can use across your learning, and hopefully you can transfer these tools to different parts of your journey of mastering complex things. But it is beyond just like using these 4 things and then just calling it a day. So we're going to go through each of them. Let's talk about the timelines that will require for each of the phases, and then I'll quickly just walk through how exactly we want to develop mastery over this complex topic here of molecular neuroscience. Okay, so when it comes to phase one, existing knowledge, what we want to do is we want to be very intentional with leveraging off of what we already know. So if someone in molecular neuroscience was reading this, they already have like a big advantage because, they've got prior knowledge. Maybe they did research on a like parallel field. And so they know, like it'll be easy for them. But for us, it's not that easy. But still, we can try making it a bit easier by using prior knowledge a lot more in terms of as a strategy. So let's actually annotate this. So here we go. Prior knowledge. And there's a lot of ways that we can do this, but one of the ways we can do this through a note-taking medium, it's a technique that we call blind mapping. And I'll demonstrate what that looks like. But this is not the only way to actually get prior knowledge or activate prior knowledge per se. Sometimes it can be useful to even try teaching what you already know. Sometimes it can be useful to even deliberately ask yourself, how does this content relate to, what I already know? So we'll try demonstrating that. By the way, I'm not an expert in this at all. So I'm in the same boat as you if you don't know anything about molecular neuroscience. The second phase is then going to be trying to evaluate existing patterns of thinking. So if phase one is existing knowledge, we'll be asking ourselves the question, how does this already relate to what I know? Phase two is evaluating existing patterns. Like, how does this relate to how I think? So I'll give you an example. This is molecular neuroscience, and this type of research, I'm not familiar with this type of research. When I read research, I'm usually reading research in learning psychology. I'm usually not reading individual studies. I'm usually reading systematic reviews or meta-analyses just because I just need like a broader reading. I'm not familiar with research methods in like a molecular neuroscience or even a clinical level at that point. And even research as a whole, like I'm not like super into research. But again, existing patterns would be something that we talk about in parallel domains. So it doesn't actually have to be about the domain itself. For example, maybe I've gotten to the hobby of making coffee. So maybe there's things that I've learned about in my hobby of making coffee that I could try applying here. At work, I have to often work with AI or learn a little bit about coding and shipping code, etc. So it may be something around like a coding kind of thing that I can do here. So what we're doing for phase two is we are trying to draw analogies and draw ways of thinking that we already do in our respective domains and try seeing if there's analogies that we can create, if there's certain ways of thinking. Obviously, someone that already has like a research background would have a relatively easier time being able to like trying to identify, okay, cool, I am a social psychology student, therefore we do research, we don't really do RCTs, we do more case studies and observational studies, longitudinal studies, therefore, it doesn't really seem like that's what's being done here. Like it's a bit easier for them, but obviously if you're not in an academia sense, it's now a little harder to get a specific understanding of this here. So that's something to mention, but it's still possible because maybe when you read through this as someone who likes to code, or even as an entrepreneur, as you like learn about business or customer psychology, maybe there's some stuff that relates there, and I'll demonstrate what that looks like as well. The third phase is going to be foreign knowledge, and this is where you'll be spending a lot of your time in terms of developing expertise. Foreign knowledge is when you have like knowledge or keywords or concepts that you don't know at all. Like you just don't know the keyword. You've never heard about it before. And again, you are acquiring new knowledge in this sense here. So there's a process in terms of how we would actually go by learning that. But the idea is that the reason we're doing a phased approach is because if we can actually start off with existing knowledge and then existing patterns of thinking, we can then use that foreign knowledge in a way to just compare. How does that fit to our existing knowledge? How does that potentially leverage off of the way that we already think? A lot of people, what actually happens when they try developing expertise with these complex topics, they go right into this process. And I mentioned for the reason that they think that there's no valuable prior knowledge or prior ways of thinking that they have. And that's a false assumption. It's a false judgment. Anytime I do calls with my students, they will start the conversation saying, Tanzeer, I have no prior knowledge. And I just ask them, just think. I need one thing. If we need to spend the next 50 minutes of this meeting for you to just think of something, we'll do that. and then they're able to always produce something. So this is possible. We're able to do phase 1 and phase 2, which makes phase 3 slightly, ever so slightly easier, but by no means does it make it extremely easy, right? There's still going to be cognitive load involved. It's still going to be overwhelming. It's a new topic. It's a meaningful goal. That's why you're doing it. Developing expertise is not easy. Otherwise, everyone would be an expert. And by definition, expertise does suggest a level of difficulty and persistence to get to, right? And the last phase is foreign patterns. So this is actually now developing the way to actually think about the expert in the specific domain. So how Lucas or Martin or Sophia, how did they think like very specifically to be able to create this type of paper in the first place? You know, what do they even do? So that's what I'm trying to think about here. Now, foreign knowledge here is going to be facilitated by a process that we can use called scaffolding or nonlinear note-taking. There is going to be an element of the best way for me to say it is going to be a mix of higher order learning and lower order learning. What this means is higher order learning, there's going to be ways to conceptually learn the information, which is what this video is going to cover. Lower order learning is actually really memorizing those specific details. So again, something about like developing expertise is that you start to learn the very, very specific things that most people don't know about a topic, right? So, you know, like if I had to talk about like a very specific thing that I know about learning science that maybe people don't know, is a concept called the misinterpreted effort hypothesis, which is basically a lot of people misinterpret effort in the learning process as bad. Therefore, when they encounter effort, they actually avoid the effort, but the effort's actually good for you, right? Another small like detail might be, you know, Atkins and Schriffen, who were the two researchers behind the multi-store model of memory, for example, right? And, you know, the year it was published, for example, if I decide to mention the year, or if I mention research on the amount of chunks that your brain is able to remember at a time, it's, you know, there's research on the magical number is 7 plus minus 2. Some more latest research say 4 chunks at a time. So that's just me kind of like showing you there's like very, very specific details that I would know about something like learning psychology, which is what makes me like somewhat of an expert. I don't really consider myself an expert. I'm still developing expertise. Maybe I'll come back in 10 years and then finally declare that. But that's what the lower order learning is. That's like the details. And that's what people usually hedge on, because that's what you can see. But the most important thing is that it's the higher order learning that you don't see that actually does a lot of the heavy lifting, which is what I'm trying to focus on with, you know, phase 1 and 2. And finally, phase 4, which is foreign patterns. This is something that you develop across months, if not years. You actually need to spend time with experts to be able to really get like a deep understanding. So in this situation, if I'm trying to become an expert on molecular neuroscience, I'm going to have to do like a unit on molecular neuroscience. I'm going to have to, like, the easiest way to do that was enroll into like a university that allows me to then like enroll in electives or units that teach molecular neuroscience, and potentially being able to get into a lab and be able to, you know, actually spend time with people who are at the forefront of this research, going to seminars, going to workshops, going to, you know, conventions that are actually doing this as well. Let me just focus the camera again. So that's the actual process that we need to engage in for foreign patterns. You know, I kind of gave you an example. For existing patterns, we're using analogies, but for foreign patterns, we're no longer using analogies. We're actually thinking about it the way that it is meant to be thought about. And that's eventually when it becomes like easy. It's just like, yeah, that just kind of makes sense. So that's what we're trying to develop. The way that I can tell you about it is that in my practice anyway, when it comes to evaluating patterns, for me, it's very instant to think about, for example, for skills development, there's a rank of technique accuracy, technique consistency, technique difficulty, technique speed. Like that pattern, I can just summon immediately. And then I'd talk about, higher order learning and lower order learning and the components of higher order learning and lower order learning. That's easy for me to think about. I think about learning enablers, and then I'd think about learning skills themselves. I think about deep processing, self-regulation, self-management, retrieval, and self-management in that sense, where it's a mindset, sorry, in that sense where it's like there's patterns of thinking I've developed in learning psychology, in coaching students on learning how to learn, that has taken years, right? Like one study session has not gotten me here. So that's important to remember for phase 3 and 4. Even though most of your journey is spent there, and inevitably because you're going to be spending a long time there, it's important to decide how you spend that time, which is what I'm trying to get to in this video. So let's do this. Let's stop explaining the theory per se. Let's go more into the practical way that we would actually go through a research paper like this, and how we would try just exploring that research paper and navigating it. So, first one is existing knowledge. We want to spend the time before we start exploring more research papers, if we do more lectures, etc., to make obvious what our existing knowledge is. We use a technique called blind mapping. I'm just going to get my phone and I'm going to set a timer. And essentially, what we're doing in this process is we are just quickly writing down what we kind of already know about the topic already. And the less of an expert that you are, the harder this is going to be, like really, really hard. And it's going to be tricky for us to actually, be able to create something meaningful. But that's the point. Like, we just still want to do something. So again, I'm going to mention this again. I don't know anything about molecular neuroscience to a point where it would make me an expert. This is like something I've never touched before. So what I'm going to do is I'm going to set a timer for 10 minutes. Actually, set a timer for 5 minutes. I won't need that much time. 5 minutes. I recommend to set a timer anywhere between 5 and 45 minutes. For a paper like this, because I don't have like a lot of prior knowledge, I can keep that at a shorter point, like 5-10 minutes. But if I did have decent prior knowledge, like if I was going to do a paper on learning psychology, at that point, I would actually give myself a little bit more time, because I do want to spend the time like really thinking about it a bit deeper and trying to hypothesize what this paper is going to talk about and discuss. So let's set that for 5 minutes. We're going to do phase 1, which is blind mapping. We're going to leverage all of that prior knowledge. Remember, prior knowledge is a strategy, not an attribute. We can actually do this. And I'm going to, without looking at Google, by the way, we're not looking at Google, in green, I'm just going to take a look. Temporal lobe. All right, cool. I'm already thinking about the brain. I mean, it's on molecular neuroscience. Obviously, it's on the brain. And the brain has different parts. And the parts do different, functions. Right? Like, that's a pattern of thinking already that I'm thinking about, right? So, that's one thing. And then we've got the temporal lobe. So, I'm just going to put temporal lobe. From what I remember, temporal lobe helps with speech. Again, I'm familiar with learning psychology, but cognitive psychology and like the parts of the brain, learning psychology doesn't really care about like specifically the anatomy. It doesn't really do that. It just cares more about the process of retention, etc. So yeah, that's what I remember. It's like good for speech. And I remember epilepsy is essentially like, it's like seizures, and seizures is like, I just kind of remember it to be like headaches. Right? Or I remember it to be like something with like malfunctioning electrical activity in the brain. This is something that I kind of remember it to be. And epilepsy is like uncontrollable as well. Right? So this is everything that I kind of know so far. We've got 3.5 minutes left. And then I have no idea what neuroinflammation is. I kind of know what inflammation is. Like if you bruise, like yourself, like the part, I don't know, gets inflamed. So I'm going to guess that it's maybe got something to do with like blood or something. So neuroinflammation, I'm going to guess it has to do something with blood. I'm going to guess it has to do something with trauma, maybe. I don't know. Maybe something with like kind of puffing up. Neuroinflammation, no idea. Maybe it's those things. Anyway, and then I have no idea what extracellular matrix remodeling is. I'm not even going to... Extracellular matrix remodeling, I have no idea. And synaptic protein alterations, no idea at all either. But I'm going to guess. I think synaptic protein alterations has got to do with something with like, I don't know, memory loss or something, maybe, or like, there's proteins in the brain and it changes it up. And then we've got something here about like extracellular matrix remodeling. No idea at all. I think it might have to do something with the same thing, like these are bad things. Now, I'm going to quickly connect these. So, one thing that I'm seeing here is that there's obviously the region of the brain. Another thing that I'm seeing here is obviously there's something here too. It seems like synaptic. I'm just going to group these as the same. I'm just going to say this as like change in brain, and I might put this as like flame in brain. I don't know. And then obviously this is like the actual seizure themselves or epilepsy that occurs. And what was happening, from what I understand, is that we're going to be looking at a certain region of the brain. That's the first thing that comes to my mind. And we're going to take a look at what happens when epilepsy occurs. So increased electrical activity happens there to a point which is detrimental. And with that, we will actually figure out that there's flaming that happens in the brain. But there's also changes of like certain things that occur. Again, dude, I'm just guessing. I'm just fluffing, if you could say. So there's 50 seconds left. I might actually quickly try cleaning this up before I move on, but this is me using prior knowledge as a strategy. It's an action. I could justify myself. I could be like, I don't know anything, oh, woe to me, therefore I need to give up. No, I'm just going to keep going. I'm just going to try using that prior knowledge as best as I can. So we've got the region of the brain that we're studying, and this region of the brain, what happens is that it actually gets susceptible to epilepsy. Here we go. And what happens here is that epilepsy then causes flame in the brain and change in the brain. Yeah, and these are both not good. Like, I think they're really bad. Yeah. Why is this even important? Like, that's another question that I might ask myself. I think it might be important because maybe before psychologists, whatever you want to call them, psychiatry, whatever it is, etc. Maybe, I'm not sure actually. Maybe they don't know about how temporal lobe epilepsy causes these things. Maybe there's not a lot of clarity on what happens at a molecular level. I think that might be the big thing, and therefore there's not a lot of clarity on like how medication might work, for example. So I think that might be the main thing here, where the reason it's important is because it goes back to research like cause and effect, I guess you could say, but maybe it's like important for medication purposes. It helps us identify specifically what's happening. Maybe it then allows us to happen what's happening at a micro level. Maybe like seizures aren't actually very well known. It's able to isolate the causes of certain things. Maybe people didn't know why neuroinflammation occurred, and or maybe extracellular matrix remodeling and synaptic protein alterations occur, and we didn't really know that before. So it's like about making the unknown known. Maybe that's why this thing is important. So again, I'm trying to justify why this could be relevant in the first place. Why does this research even matter? So that's my understanding of things. You can see, not very sophisticated, but it's a good starting point nonetheless. At least I have something. It's easier to build off a faulty schema than it is no schema. Now, my timer is up, but what I might do now is I'm going to just move on to phase 2. This is still within our priming phase, by the way, like things that we will do right before we actually get into the paper itself. Phase 2 is going to be evaluating patterns. Now I want to try creating an analogy or try thinking about where I've seen this type of research before. So, or I've seen this type of pattern before. I'm going to give a really silly example. Like, when it came to cigarettes, right? People always thought like cigarettes were good for you, but eventually when research was done, smoking was associated with lung cancer, increased rate of depression, for example, and halitosis. I don't know, bad breath. Nah, that's not a good reason. But it was definitely like, your lungs would get black, you would get lung cancer, whatnot. Um, so, for a while, there wasn't any research on that. Maybe that's like an analogy. It's not the perfect analogy because something like temporal lobe epilepsy, it's like uncontrollable. It's something that happens in the body. So it's something that and like is associated with neuroinflammation, extracellular matrix remodeling, and synaptic alterations. I don't know. Those things seem to be like something in molecular, right? Molecular, something that happens at like a micro level, and therefore maybe that's something that needs to be done there. Uh, okay. Um, so I don't know. What's like something that could fit here as an analogy? This process is hard, by the way. So if when you do it and you realize it's hard, that's good. This is your brain thinking a little bit. Maybe something like AI. I don't know. This is coming to my mind. There was actually research done by MIT labs and even emerging research that talks about how AI use is actually associated with poorer learning outcomes, poorer retention, and reduced cognitive load and uh, just sustained effort during the learning process. I mean, that's just generic, right? Cause and effect. And the use of AI is not something that's uncontrollable. So, I don't know. This is hard. That's like the best analogy I can kind of think of when it comes to temporal lobe epilepsy is being associated with these things. Like maybe we didn't really know how AI influenced us, and now we kind of know a little bit more about it and what happens at a cognitive level. But again, you can see because I don't have like a very, very specific schema or awareness of how things in molecular neuroscience occur, or even research in like clinical fields, this is definitely very hard for me. But that's the point. I'm trying to think about existing ways of how I think and apply that there. I might just use that as an example, to be honest. So eventually we realized that AI was reducing cognitive load during the process. It was reducing learning outcomes. It was also reducing retention, right? So I kind of remember reading some research on that. It kind of seems similar, maybe ish, but in this situation, obviously, it's more of in a like medical context, right? So I don't know. Yeah, I'm just gonna go for it. I don't think it's a perfect analogy because there's some differences. By the way, you see how I'm evaluating between my analogy and the reality of this by guessing it. That's that comparative thinking that's going on. That's really useful, and it's really tiring as well. So that's something I'm thinking about. The reason it's different is because temporal lobe epilepsy is like a thing that happens to humans, right? It's like something that happens to the brain. But AI is like something that we use, deliberately. So it's not a perfect fit of an analogy. Also, like it being associated with like bodily hurt, I just don't see these things being something there. Also, the scale of it as well, like cognitive load, learning outcomes, and retention, those are measured by other means. Whereas neuroinflammation, extracellular matrix remodeling, and synaptic protein alterations, I would actually like to think those are more observable. Like you'd be able to actually get a microscope or something, and then you'd be able to maybe like see that stuff. So it's not a perfect analogy, but I'm going to park it there for now because you can see that's what's making me think. So we just done two things there. We did the existing knowledge using the blind map, and that's where I got to this point here where I kind of have some knowledge around temporal lobe, frontal lobe, different parts of the brain and the functions that they have as well. I had a little bit of knowledge around what epilepsy is, not a lot. I kind of knew what seizures was, but not a lot. I could be wrong, honestly. I kind of guessed what the purpose of this research paper is. Who knows? I could be very off. And then afterwards, after we did phase 1 a little bit, phase 2 was about existing patterns. Like, how does this relate to existing patterns that I think already? And unfortunately, because a lot of my learning has been more software based, a little bit more like psychological constructs based, a little bit more on like a macro scale, a little bit more on randomized controlled trials, RCTs, not this type of study per se. This could be an RCT. I don't even know. No, I don't think it is. That would be weird. But yeah, like that. That's what I'm thinking about. Yeah, that's like a way that I'm thinking. Now, we have concluded phase 1 and 2 because I've just done this. And technically speaking, this part of the process can last longer. And what I should mention is that every time like you kind of introduce new material, you kind of go back. Now we're going to be entering phase 3 a little bit more and a little bit of phase 4 because now we've done this process, and this is actually going to be me going through this paper and trying to develop a little bit more insight into what all of these things are. But the idea is that phase 1 and 2, you want to do that every time you sit down with something, and you already do it like subconsciously. So as you start to become more of an expert in molecular neuroscience, if that's what you decide to do, you will, every time you start learning about something, you want to be very intentional about asking yourself, how does this relate to how I already think? How does this relate to what I'm thinking about, what I learned the last week? Because you don't want to lead up to chance, right? What ends up happening is when you get two people that haven't done cognitive retraining, so in this sense, like learning how to learn, the differences in their ability to learn usually comes back to how automatic this process of comparing and contrasting is. But when you're intentional about phase 1 and 2, this is that comparing and contrasting is just a natural byproduct of a simple change of just being a little bit more intentional with using prior knowledge. So again, as I mentioned towards the earlier part, if you already knew very well about this type of research, you would spend more time in phase 1 and 2. But for me, because I'm a beginner, I wouldn't spend too much time here. If you're not a beginner, it makes sense to spend a bit more time here. Nice. Let's move on to this one here, phase 3 and 4, because this is where a meat of the conversation and discussion would be. Phase 3 is really just talking about, well, now let's actually start reading this paper, because a lot of this is going to be foreign. Let's start developing a little bit of expertise around this, and let's start thinking about how things are. So what I'm going to do, and what I usually do during this process, is I just have the map open here, and I like to build on top of this map. And there's a very specific search strategy that I have. So what I need to do is I need to make sure that in phase 3, I am trying to start off with the most basic keywords, the keywords that I feel like would have high connectivity. So we want to have that, like the keywords that really define the topic. That's what we want to start off with first. And then we want to move our way on to specificity, the really specific keywords that just don't really connect to anything. So it's actually very silly if I scroll down here and I try learning a little bit more about peptide N termini and K, like TMT 16 plex. I feel like this is like a very, very, very specific, isolated. I wouldn't want to learn this foreign knowledge first. But something like temporal lobe, yeah, I kind of need to know what that is. And then focal epilepsy, I kind of need to know what that is. And hippocampal sclerosis, I need to know what that is. Neuronal cell loss, I probably might need to know what that is as well. But you can see those keywords like fit usually like here. But then specificity is what fits here. And again, as I mentioned earlier, when it comes to expertise, a lot of people think, and they can only kind of see this, and they're like, oh, if I don't get here, then I am not an expert. But that's actually not true, because experts spent years developing this stuff like really well, and that's what makes a specific thing a little easier to learn. As I said, it's not going to make it like Very, very easy to learn. It just makes it a bit easier to learn. And depending on the level of expertise that you're trying to go through, you will need to commit things through rote memorization if you want to develop expertise like very, very, very quickly. But if you're trying to develop it in a more organic way, which is what usually happens in something like research, you won't need to do like flashcards and you won't need to have like a lower order learning strategy. It's because, you know, the process of reading research and just doing it enough times and writing it and just going through like journals and like constantly just revising, you know, your paper and getting it reviewed and getting destroyed by whoever, you know, you're getting, like your professor, whatever it is, that usually already just makes it a lot easier to learn these details in the first place. So that's what we want to do for this map here. We want to start with those broad keywords and then we want to slowly make our way to the specific keywords. So let's get into it. Temporal lobe epilepsy. I'm just going to read a little bit of this, but even before I read this, I don't even know what this topic is about. I don't even know what temporal lobe epilepsy is. So what I'm going to do is I'm just going to copy paste that. I'm going to go to Google. I'm going to skip the AI overview, by the way, a little bit, and then I'm just going to quickly go into images. Just makes it a bit easier for me to process the information. There's a bit of emerging research that shows that your brain processes visual information a lot faster than written information, so this is why it's useful. So temporal lobe, it seems to be, you know, the big part, like that middle part of your brain. Okay, cool. And there seems to be different things for this. So epilepsy symptoms based on affected lobe. So there's something there. I might need to learn a little bit more about the temporal lobe. So I can just quickly click that. I'll just go into images really quickly before I go into the AI overview. And nice. Let's take a look if there's an image. Here we go. This is a nice image that quickly explains what the temporal lobe is. So temporal lobe is one of the 5 lobes in your brain. So it manages or contributes to several of your most useful abilities, such as object and face recognition, language, understanding expression, memory recall, and feelings, like your emotions. So when I read this, I mean, this is new information to me. I didn't actually know this. I mean, I did say something about how it's like related to speech, but that's only one part of this. Okay. So now that I've read this is new information coming in. What I want to do is I want to kind of connect it and make it easy. I don't want to make it too overwhelming. When I read this, it's super overwhelming. But when I read this is a bit more manageable. So when I see this, I see that temporal lobe is responsible for facts, feelings. That's something that pops up to me. And object facial recognition and like speaking, like seeing and speaking. Those are 4 really kind of cool ways, like seeing, speaking, and then facts, feelings. So what I might do is I might just go back to my notes and I might just write this. So I'm writing temporal lobe as one of the parts of your brain, right? I could actually just do that as well. Brain lobes. There's like 5 brain lobes and this is one of them. I'll just put that there. Why not? And it seems like there's 4 things it's useful for. Number 1, seeing, and we're not seeing specifically because seeing is actually the occipital lobe, so that's too simple. Object and facial recognition. Okay. All right. So faces and speech, and then facts, feelings. There we go. So now one might argue, Tanzeer, this is actually not what's written. Here it's written memory, storage, and recall. Here it's written emotional reactions and processing. Here it's written object and facial recognition. Here it's written language, understanding, and expression. For me, I want to reduce the complexity. I don't care because when I think about face, I'll think about facial recognition and object recognition. When I think about speech, yeah, language understanding. Like this, if I need to learn the specifics for this, I can do that later. I'm just in that process where I want to get a basic understanding. So I don't want to get caught up on the specific keywords. I just want to make it make sense to me. I'm the judge of what I think is useful for me right now. And that's what I think is useful for me. So that's what the temporal lobe is. It's facts, face, speech, facts, feelings. That's what it's useful for. Now, I might need to learn a little bit more about epilepsy because I don't know what that is. If I just take a look at images. Epilepsy is long-term brain condition with repeated seizures. I don't know what a seizure is. Seizure is a sudden, uncontrolled surge of electrical activity in the brain. It temporarily disrupts how cells communicate, leading to sudden changes. Oh, okay, interesting. All right. So, what can happen in the temporal lobe is that, I mean, it seems like a seizure kind of happens and then it goes to the temporal lobe. Or is it that, like, the temporal lobe is where it starts? Sudden bursts of electrical activity in the brain. So a question that I have is like, where does a seizure start? Does it start from like a different part of the body, or does it start within the brain itself? But when I read that, that's pretty interesting. So how does it connect? I mean, epilepsy, I don't really have a lot of specificity here. So temporal lobe, what will happen is that usually there's like, I mean, in the brain, there's electricity. I kind of already know that. And there's neurons. And the temporal lobe, it's a bunch of neurons. And what can happen in the temporal lobe is there can be a seizure, which is like uncontrolled like there's just too much electricity, too much zap, right? That can actually occur in the brain, and I might do this instead. I don't think temporal lobe leads into that. I think the seizure itself is like a standalone concept. And I might just Google a little, I might just take a look at the images, like temporarily disrupt how cells communicate. So there's like a cell direct thing on cells. Okay, nice. And then movement, etc. All right. Seizures don't always mean epilepsy. That's also potentially a bit important to remember if I needed a bit more specificity. Focal seizures, one area of the brain is affected. Generalized seizures, all parts. Ooh, that's important actually, because there's actually different types of them. And I remember on the paper saying that temporal lobe epilepsy is the most common type of a focal seizure. Most common form of focal epilepsy. Okay. Yeah, cool. Nice. So, seizure is that. Obviously, epilepsy in that situation would be like multiple, right? This is like one, and this is many. Is that how epilepsy is defined? Multiple, right? Long term. It's long term. It's long term. So this might be like short term. This might be long term. Nice. And each of them, they've got like their areas. So something else that I'm learning a little bit more about is like, and you can see, by the way, you know how I put short term and long term here, and I put one to many here? This is prior knowledge kicking in, because this is me making that split of how I think and my patterns of thinking, right? So we're really leaning on that. So when it comes to seizures, there's also something to do with generalized versus focal. Generalized versus focal. And basically, that is like contingent on whether it's the whole brain or like a specific part of the brain, right? So whole or part. And for those watching, there's a program technique we call whole part whole. It's a technique that we teach. It's also kind of known in teaching as well, so that's quite easy for me to remember as well. Whole part. Again, I'm leaning on like prior knowledge, analogies, ways of thinking that I already have, and I'm integrating those into my notes because it's way easier to remember those things. And so what actually happens is that we know that when it comes to these seizures, the epilepsy here per se, and there's general and there's focal, general and focal epilepsy in this situation, from what I can recall. The most common types of this is temporal lobe seizures. Most common type of focal epilepsy. Nice. Sweet. And the way that I might draw this is I might just draw it like this, where it's like
we've got epilepsy as one like key concept being talked about here, and then temporal lobe being another key concept, and a result of these two coming together is like temporal lobe seizures. No, not temporal lobe seizures, temporal lobe epilepsy. That's right. Nice. All right, cool. Epilepsy. Okay, so now what I might do slowly is this is where I'd actually integrate excuse me, I'd integrate certain techniques. So phase 4 is when we start developing different ways and patterns of thinking, and I talked about interleaving here. So a technique or a strategy that I usually like to teach a few of my students is something called micro teaching, where as you actually learn, you want to teach it a little bit. So if we go back to that paper, when I read temporal lobe epilepsy, I had no idea what it was, but now I can kind of explain it. So basically, you've got your brain has many neurons, and these neurons, the way that they communicate with one another is sending like tiny bits of electrical activity with one another. But what can happen sometimes, unfortunately, is that the electrical activity can be a bit like too much, and out of nowhere, it's like way too much. And that actually interacts and interferes with the way that your brain operates and the way that your cells communicate to one another in your brain. And that can lead to many differences. That can lead to problems in your smelling, it can lead to problems in your movement, it can lead to problems in your mood, it can lead to many, many things. And what actually happens is that some people, they get seizures a lot, long term, and we call these epilepsy, where people might have random seizures that occur across. Epilepsy, there's quite a few ways that it happens. One time it can happen on just across the whole brain, like a seizure can happen across the whole brain, but sometimes a seizure might happen in a very specific part of the brain. When it happens across the whole brain, it's called generalized epilepsy, but when it happens in a very, very specific part of the brain, it's called focal epilepsy. Now, there's different parts of the brain. You've got your occipital lobe, which is responsible for what you see. You've got your prefrontal cortex, which is responsible for problem solving, etc. But you've also got one part which is called the temporal lobe. And I like to say that there's 4 things that your temporal lobe is important for. Number 1, recognizing faces and objects. Number 2, remembering facts. Number 3, processing your emotions and feelings. And number 4, being able to speak as well. So when you get epilepsy and you get a seizure, it can happen either across the brain or a very specific part of the brain. And what can happen is that it can actually it can attack or occur as a focal epilepsy, as a focal seizure in the temporal lobe. And this can then potentially actually cause people to slur their words. It can cause them to forget information. It can cause them to not process information correctly, or actually feel a certain way because it hijacks the emotions. And this is what we call temporal lobe epilepsy. So you can see what I've done there. It's taken a little bit to get to that point, but I'm now able to understand that. Nice. But I have no idea what neuroinflammation, extracellular matrix remodeling, and synaptic protein alterations are. And that's what we're going to do. We're going to figure out what that is, because that's what's happening here. But what I might do is I might try confirming if my understanding is correct in that sense. So, most common form of focal epilepsy characterized by recurring seizures. I already know what that means now. Nice. I know what focal epilepsy is. These seizures often cause a mix of unique sensory, emotional, and cognitive symptoms. They are treated with anti-seizure medications or surgery. So, auras. That's funny. You get auras. Okay. Wow, that's actually quite scary, actually. Deja vu, impaired awareness, automatisms. Okay. Like, alright, I think I've gotten a little bit of an understanding of what this is. Yep, most common. Start in one or both temporal lobes. Oh, there's 2. Yeah, that makes sense. Okay, and then surgery may help people with TLE become free. What causes TLE? But what causes it? I don't understand. Yeah, but prior brain trauma, genetic disposition. Okay, I see. So, all right, nice. Okay, well, let's move on because I think I've got like a working understanding of that. Now, neuroinflammation. Let's learn a little bit more about what neuroinflammation is. So, let's go to images. Okay, it looks like if you get injured, yeah, that's kind of like my prediction of what neuroinflammation might be. Like if you have some, but then there's this new keyword, microglia, microglia, however you say it, I don't know. There's neurons that you have. There's these different parts of the brain as well. Okay. So you can see I'm skipping through because I just want to not be too overwhelmed. I just want to create some in me. I hope it doesn't. Yeah. Okay. So I'm not getting a strong understanding of what it is yet based on the images. So I might write the complex inflammatory response of the central nervous system mediated by immune cells and astrocytes, while initially protective against injury or infection. Chronic neuroinflammation damages healthy cells, accelerating neurodegenerative diseases. So it seems like, yeah, okay, this is a good photo. Neuroinflammation helps protect the brain by fighting off infections and aiding the healing process. So that tells me what it does, but I don't know what it specifically is yet. It, like, releasing cytokines and pathogens to aid tissue repair. Okay, so it's like, so neuroinflammation is like a immune system response. So if I had to guess, like neuroinflammation, yeah, that like I kind of remember my studies from genetics, or not genetics, immune system studies. So it's like part of the immune system, yes, and it protects against injury or diseases. But the specific mechanism, like what is it? Is it like is it like, does your cells like increase or something? Like, is it just more blood flow or something, or does it get warmer? So this is where I might Google just like what actually is it? Inflammatory chemicals. Okay, but what's that? Molecules released by your immune system to help. Okay, nice. Yeah. So like molecules. It's actually just molecules released to fight. Yeah, cool. Maybe there's a nice image that shows this. I don't know what that is. Because I kind of know what inflammation is. There's like a bunch of blood coming through. Whatever. I guess it's enough for a working understanding. Like, your brain releases. I didn't know you know this at all. Widen blood vessels. Yeah, that's what I need to know. Their job is to widen blood vessels, increase blood flow. Yep, and guide white blood cells to the area to fly. Yes, that's what I remember inflammation being, right? Like, so there's like increased blood flow, right? And widen blood vessels. Yep. And yeah, and like white blood cells go to this area. Okay, cool. So that's okay. That kind of gives me like a little bit of a working understanding of what it is. And yeah, like, so like, what does this manifest as? I'm just trying to think. Like, when I injure when I like when I hurt my hand, it like fuels up, right? Like, that part of my body, and like I think more blood just goes there, and like maybe more blood just helps it heal a little bit more. I don't know the specific mechanism. Clearly you can tell I'm just really dumb, but yeah, maybe this is just kind of like that, whatever. Okay, I feel like I got a working understanding. Nice. Like, maybe if I hurt my head, and like, obviously inside, some things happen, some things move around, or like there's potential trauma. My body needs to like send more blood there so it can like form scabs, and so it can stop the bleeding. Yeah, nice, whatever, cool. Let's go to this really complex thing. Come on, man, no. uh Neuroscience. In neuroscience, ECM is the dynamic activity dependent breakdown, synthesis, and recycling of structural and functional molecules in the brain. I have no idea what even in the world that is. I'm going to skip. That's a bit hard. I'm going to get back to it later. But what I know so far is that temporal lobe epilepsy is associated with neuroinflammation. So basically, it's saying that seizures, specifically temporal lobe epilepsy, is associated with releasing or like triggering neuroinflammation, which might be useful because I don't know if it was that was known in the past. Therefore, maybe they can create medication that's used for neuroinflammation for temporal lobe epilepsy. Or if there's known surgery that reduces neuroinflammation in the brain, maybe that helps. I don't know. Anyway, I have no idea what that ECM thing is. I'm going to skip that because that is going to hurt my brain. I'll get back to it later. It's not me avoiding cognitive load. It's just maybe it is me avoiding cognitive. Now, I'm just trying to think a little bit more, right? Synaptic protein alterations refer to changes in composition, abundance of functions of protein in a synapse. Sorry, pal. No idea. Pal, I got no idea. What even is this? I'm gonna try. Refers to changes in the composition, abundance, or function of proteins at a synapse. Okay, so this is introducing a few new key terms, especially that ECM one as well that I was reading. Let's just go back to this. So this is me having to still develop like a working understanding of these things. Neurology, no, neuroscience. Yeah, I think this is useful. Remodeling, breakdown, synthesis, and molecular of brain's non-cellular scaffolding. So I don't have a working understanding of this specifically yet. But that's okay, because this is what developing expertise is, right? It's going to be that challenging process. So let's go through a little bit of this. Synaptic protein alterations. What is this? Changes in composition, abundance, or functions of proteins at a synapse. Or synapse, I don't know how you say that. What even is this? I don't know what this is yet. Specialized junction where a nerve communicates to another one. Oh, cool. That's a bit easier to remember. Oh, is it nerve or neuron? Yeah. Oh, yeah. I remember this stuff. It's a specialized junction where neurons communicate with one another. It allows electrical or chemical signals to travel between nerve cells, muscles, and glands. Yeah. Okay. So if we just try mapping things out a little bit more, neurons. Now, I know what a neuron is, kind of. It's like your brain cell that I lack. And neurons get connected to other neurons through like a synapse, which is kind of like a middleman. So again, this is me trying to create like analogies. So if a neuron is like a road, a synapse is a bridge connecting those roads together. So this is like bridges. And then there's something around, okay, that kind of makes sense. And it allows chemical, let me just get the camera working again. It allows chemical and electrical signals to travel with one another. It just allows you to move, That's maybe what it is. Yeah. Anyway, let's get into a little bit more of it. So that's something you was saying, and basically S and P. That makes a little bit more sense. Synaptic refers to the location. Okay. Protein. Okay. So proteins at a synapse. Proteins at a synapse. What even is a protein? I actually don't know what a protein is. All I know is like you got to eat a lot if you want to get huge. But like other than that, I actually don't know. Fundamental machinery of a synapse driving neurotransmitter release and signal. They are concentrated in three zones. Are responsible for packaging vesicles. So it kind of seems like proteins allow the transferring to happen. Are the fundamental machinery of. Okay. All right. Yeah. That kind of seems like protein is like the glue or like the catalyst. Whatever. Okay, I'm just gonna guess. I'm just taking a wild guess here that protein is just the catalyst that allows that to happen, and so as synaptic protein alterations. So I'm developing working understanding. Synaptic is the location, so that's the bridges between the neurons. Protein is the actual thing that allows a synapse, like the synapse, whatever however you say it, to do like the thing that's meant to do. And then alterations refers to changes. Change in the composition, abundance, or function. These molecular shifts disrupt neural communication and plasticity and are heavily implicated. Ah, okay, interesting. So synaptic protein alterations is when the protein itself like changes. In this sense here, it seems like even if it changes in like a whatever direction, the shift disrupts the way that the brain communicates, and that can lead to like neurodevelopmental, neurodegenerative. Okay, I see. Okay, that gives me a little bit of a working understanding of what this is now. Nice. So it seems like the neuron in this situation was an important concept to revisit. I think my iPad's a bit frozen a little bit. Here we go. So this is essentially about that makes a little bit more sense now. So protein, and basically what happens is that a change here, so I could actually have this in another color, maybe like a change here is like really bad because that reduces the how the like the neural communication happens. Yep. It reduces the plasticity, and I think a big idea is that it can cause those diseases. I'm just going to call it neurodisease. I know that's not like a real word, but I don't have to do neurodegenerative, neurodevelopmental, and all that. It seems like that's what it's like causing, like Alzheimer's and whatnot. Interesting. Okay, cool. So synaptic protein alterations makes a bit of sense now. Nice. How does that relate to something like neuroinflammation? No idea. Do I want to find out? Maybe? Maybe is it neuroinflammation that leads into synaptic protein alterations? Is it the fact that there's so much like the immune response is occurring, and then that's happening? Is it not related at all? Are they parallel? Does neuroinflammation happen first, and then synaptic protein alterations happen afterwards? I don't know, and these might be questions that I ask if I had access, and like an expert might actually know that question, like answer the question very well. I don't know it, but I'll be honest. I think that's a really good question to be able to ask, right? To be able to get to this point where we're asking this type of question, that's really good. Now, I just need to learn a little bit more about this extracellular matrix remodeling in neuroscience. So I just don't know enough about that just yet. Okay, so here it's about involves the dynamic degradation, synthesis, and molecular recycling of non-cellular scaffolding in the brain. I think I have a neurodegenerative disorder as I read that. I have no idea. But that's the beauty of learning. You get to find out. Let's find out before, obviously, we just wrap up this part of the demonstration. So let's take a look here. Uh, okay, made a comment, which is useful. I'm overwhelmed right now. Like when I see this page, it's like, dude, I know nothing. It's kind of exciting. It's kind of overwhelming. But the point is, especially in phases, like especially phase 3 foreign knowledge, especially because we're starting phase 3.1, which is basic knowledge, we do want to make it less overwhelming. And this mapping has made it very less, like, not as overwhelming. Let's go a little bit here. So let's take a look. Why am I pretending when I look at these pictures, I actually understand what's going on? I'm just gonna be honest. I need to stop doing this because I have no idea. Okay, let's try seeing what is extracellular matrix. Okay, ooh, this is a what's it? A holy refresh pool. You know how they say that when you search like new topics. This is so Gen Z, but this is really good. So I was able to isolate that. ECM is a complex non-cellular 3D network of proteins, carbohydrates, and minerals. It surrounds cells and tissues, providing essential structure scaffolding while driving critical biochemical signals. So, what does non-cellular mean? Oh, it's just one big thing, huh? Ah, so cells, yeah, I kind of remember like cells can like reproduce and then like fundamental building blocks, but some like non-cellular means it's just not organized as a cell. Yeah, okay. So non-cellular is like another keyword. So it seems like protein are protein cells? No, they're not, right? Are they made up of cells? Oh, okay. Proteins live inside of cells. Dude, I've just forgotten what a cell is, man. Like, okay, whatever. Yeah, okay. So yeah, this is relating to the cell, and obviously the protein is the thing that's within the cell. So this is like synapse protein alterations looks like a cell, and whatever we're talking about here, extracellular, it's acellular. Is that a word? So do you see how I'm like doing this, like cellular versus acellular? It's not necessarily mentioned as like two different ways of delineating between these two, but I decide that, right? That's cool. So extracellular ECM a complex non-cellular 3D network of proteins. So yeah, proteins, carbohydrates, and minerals, and it surrounds the tissues, providing a structure. Yeah, that's all I know. Like cellular glue participant. It drives the tissue shape. Nice. So it's actually like a nurse kind of thing, or like even more than like a defense, but also like a catalyst as well. So extracellular matrix is okay. Let me look at the images. I might be able to pretend that I know something. Okay. Let's take a look. I definitely don't know anything that's going on. All right, but I think I have a working understanding. Um, so it's like protein, carbohydrates, and minerals. I don't know, and I don't like. I actually don't know what each of those are. I just know I eat them every day, like carbs, minerals, and I know they're like important for you. But yeah, I have spent the time to actually learn about them. I should do that one day. Maybe the day is today. Okay. Non-cellular component present within all tissues and organs. scaffolding for the... Okay, so scaffolding is like, it like shapes it. That's what I think it is, right? Like it helps shape the thing. And so when you get modeling, it's a breakdown of that shape, right? Remodeling refers to the dynamic breakdown, synthesis, and modification of the brain's structure, and it's essential for regulating synaptic plasticity. So that's really important. ECM would actually lead into synaptic plasticity because that's what the synaptic protein is important for, and it's essential for regulating, controlling iron homeostasis and determining brain health. Cool. But what does remodeling mean? It means the breakdown. It means changing. Is that a bad thing? Is that a good thing? Okay, let's take a look at nature.com. What are they saying? What do they define ECM as, huh? And the C is... Oh, maybe this paper talks about it. Yeah, I can definitely reference this one, by the way, because, I mean, we're trying to learn this one in the first step. Is this a good thing? Like, good and essential, but it becomes harmful when it becomes dysregulated. Ah, okay, cool. So all I know is that it changes, like the structure, and it can be good or bad. And it can be bad sometimes when it doesn't work in that direction. Cool. So this is going to be where I've gotten up to in almost an hour. So you can see it does take time, but we just did a little bit of that phase 3, a tiny bit of phase 4 as well. Phase 4 is really largely dependent on me then sharing like these notes with an expert and getting beat up and them calling me dumb and me learning a little more. But you can see like we're still here, like in my opinion, because there's still so much that needs to be done. Like I haven't even read this paper yet, right? But there is so much essential knowledge that I just do not have where I don't even deserve to read this paper. Like I wouldn't know anything that's going on. But at least now I would actually agree. I would argue that I kind of already know I kind of reading the title. I understand the title now. Yay. And now I can get into the abstract and learn a little bit more. So if I read the introduction, temporal lobe epilepsy, which are what I kind of already know, is the most prevalent form of drug-resistant focal epilepsy. Okay, so focal epilepsy I know, and is frequently associated with these things. Okay, cool. I haven't learned about them yet, but these seem like bad things. Right? Neurocenal cell loss doesn't really seem really good. And astrogliosis also seems like a bad thing. I can quickly search that now. Defensive response. Okay, cool. Yeah, and I kind of learnt about one of those defensive responses. And so now when I search up that, when I search up this keyword, this is actually a good kind of demonstration. I've actually got neuroinflammation as like a comparison point. Like, how is this different from neuroinflammation? Or does this cause neuroinflammation? Or does this happen somewhere else in neuroinflammation? Yeah, like what even is the difference? Oh, it's a broad. Okay, all right. So now I understand that it's a subset of neuroinflammation, and that type of question that I was able to ask was very powerful. And what I would then do is I would actually commit to more learning, and if I did that, this video would take ages to finish, and it's already getting relatively long. So here I'm going to learn a little bit more about. In this study, we performed mass spectrometry-based proteomic profiling of microdissected hippocampal. Yep. Okay. So from what I read, they like did something with a microscope, and they took like samples from people that have TLE, and yeah, how, dude? They just took part of the brain. White tissue matter. Really? Like. Okay, I have no idea how that. How do they take that from their brain? Dude, like, Okay, well, I might have to figure that out later. But yeah, I'd learn more about what hippocampal tissue is, what TLE tissue is hippocampal TLE tissue, hippocampal temporal lobe epileptic tissue. Like, is that what it's saying here? Because that's what TLE is mentioned as here. So I would actually learn a little bit more about these keywords. And the cool thing is that now I've actually got this face, like this foundation, and this type of paper is going to be something that is going to take me weeks, take me days to actually understand. And that's okay. And this foundation largely happened because we were relying on prior knowledge. We were being quite patient with trying to figure out trends. And this is how we slowly develop the process. If in future videos, we might be able to dive into phase 3 and phase 4 a bit more. But this exact process, I just keep going back and forth and I keep reading the paper, and slowly things start to make sense.
So that paper at the start probably felt impossible, but after running that four-phase technique, you can see how much of the complexity doesn't drop itself, but the way that we're approaching that complexity is a lot more productive, and so it increases the chance that I'll be able to learn the topic. Now, it's not because I became an expert, it's because I stopped trying to absorb everything at once and started running the process that an expert's brain runs automatically. So again, you do not need to be an expert. You can't because you're trying to become an expert, but you need to think like an expert. And as you think like an expert in the early stages of developing mastery, you'll slowly develop mastery to a point where people will start calling you an expert. So try the four phase technique out on a topic that you've been avoiding or find complex, and let me know what you think in the comments. I'm responding to every single comment.